So let's go ahead and we're going to start kind of talking about our expectations for this lab. We wanted to make sure that it works well. So our learning process is all about respect. It's respect for ourselves, for each other, and for our whole world. And we ask all of you, our commenters and viewers, to show the same respect that you would in an on-site museum program. We want to make sure that everyone is using kind language and appropriate words, because this is a program for everybody, right? My friend Max will be moderating the chat for any kind of disrespectful language, but we welcome all ideas that are founded in respect and invite everyone to do the same in this online space. And we have Annika joining us. Wonderful, wonderful. So the lab at the museum, right, is a physical space, and we're kind of doing this lab in a virtual space. And it's going to be different than a regular lab, but we're going to try to keep some things the same. What I'm really hoping is that I can still ask you guys lots of questions and that you can ask me lots of questions. I know it's not quite the same for typing as opposed to talking, but I would love to hear from you guys, hear what interests you, hear your ideas. A couple of my experiments today, I'm going to ask you guys to choose. So I'll tell you more about that when we get to it. But what we're going to do is we're going to start off talking about kind of our main idea, and then we'll start our experiment, and you guys are welcome to do it along at home. Oh, this is great. Oh, Krista is sharing that they reuse plastic bottles to save the turtles. That's really awesome. All right. You guys have all said a little bit about yourselves in the chat, which is awesome. Max is getting up our pictures because a lot of times in the lab we have, just kind of like printed out pictures, or we have an iPad that we can show you pictures. And what we're talking about today is a little hard to, it's hard to picture in your brain because it's so small. What we're going to be talking about today is DNA. Have you guys ever heard of DNA? You can put in the chat. Have you ever heard of DNA before? Does anybody know what it stands for or what that means? Any ideas about it? I'm going to give you guys just a second to see if you want to respond. So maybe some of us know about it. That's awesome. Oh, this is a comment from Olivia and Johnny about not being able to see everyone here. It's true that you'll only be able to see me and my camera. Everyone else is only able to kind of communicate through the chat. Got it. So, all right now I'm hearing that some people have heard of DNA before, and we're going to talk about kind of what DNA is, right? DNA is something that's very, very, very small. It's a molecule. Oh, I love this. Susan says, it's like the building blocks of life. I like that, right? It's kind of like a recipe, right? In a recipe, you have a list of ingredients to make something like a cake, and you have kind of the instructions for doing it. That's what DNA is. Ooh, this is a great comment from Krista. It makes you, you. Yeah. Oh, we have one from Julian. DNA is an acid, that controls everything about you. So it's true that DNA lives inside of our cells, right? DNA lives in all of the teeny tiny pieces that make up our bodies and the bodies of everything alive, right? And we're going to talk a little bit more about what it is. Let's take a look at what it kind of looks like. I'm going to have Max pull up a picture for me. First, I'm going to show you this picture. These are a couple of pictures here that show kind of like the science idea of what DNA looks like, right? So you have these kind of spirals that go back and forth. It almost looks like a ladder. When you Google pictures of DNA, it's kind of interesting. They're often, like really cool colors like blue, or sometimes they have multiple colors. But this is just kind of an image to help us understand what it looks like up close. If we are really, really taking a real picture of DNA, it's going to look kind of like our next slide. Can we take a quick look at that, Max? Let's see if we can take another look. So I'm not currently able to see it. Can you guys see it? All right, let's see if I can show you. Whoops. There we go. Beautiful. Thank you so much, Max. So if we're looking at a real picture of DNA, It looks kind of like this. This is a picture taken by an electron microscope. So it takes pictures of things that are really, really, really tiny. We can't even see them with a normal microscope. We have to have a special one that can see things that are super small. And it looks kind of like this. It looks kind of like a line with some more lines in it, right? So it doesn't look maybe as cool as some of those kind of scientific illustration depictions, but that's because it's so small. We can go ahead to our next slide here, Max. Beautiful. So this DNA is like a tangle of almost like spaghetti, right? It's this tangle of little teeny tiny molecules, little teeny tiny pieces, all strung together. And some of that string, some of that spaghetti, clumps up in a certain pattern. About 2% of our DNA, a small amount, makes up our chromosomes. And our chromosomes clump up in certain patterns as well. You can see that over here on the left, all of these little things that kind of look like almost like X's. Those are kind of more tangles of spaghetti. And those chromosomes are what have information about many, many things about us, right? As some of us have mentioned in the chat, DNA determines a lot of things about us, a lot of the ways that we look, even maybe some of the ways that we feel and act. It's sort of like the beginning, right? Our decisions and thoughts and our experiences, of course, shape who we are, but DNA has a lot to do with it. It especially has a lot to create in creating like how I look, right? The fact that I have green eyes or brown hair. If we look really, really closely at those chromosomes, we can kind of see, this is that picture on the right, how they're a little bit kind of clumped together. There's all these teeny, teeny, tiny strings. Those strings are the DNA. So we're going to go ahead and move to our last slide here, last picture, I promise. We said that DNA lives in our cells, and it's kind of weird to think that we're all made up of these teeny, teeny, tiny pieces because, this picture on the right here is some human skin cells. And, when I look at my skin, I don't see a bunch of little circles. It's because all of those little circles, those cells, are so tiny, right? I don't see the shape of the cells themselves. I don't see any of the empty space between them. It's just because they're so tiny. Over here on the left is a picture of a mouse cell, and you can see the different parts of the cell. The cell we're going to be looking at kind of this big kind of dark circle that has a little tangle in it. That dark circle is called the nucleus. It's a part of the cell, and it carries all of the DNA. So all of the DNA of this cell, this mouse cell, is really tangled up and squished into a cell inside of the nucleus of the cell of this animal. So it's really, really tiny, so tiny we can't see it. We're going to go ahead and bring our screen back here. We've had some really, really great comments in the chat. I love these, talking about how, of course, yeah, DNA is why people are different. It determines lots of things about us. It stands for, this is a great addition, deoxyribonucleic acid. And that's just kind of the name of all the little pieces, the molecules that go up, that go together to make it up. That's great. We have, let's see, Ayla saying, oh, that DNA is half from one parent and half from the other. That's true, that we do get our DNA from our parents. Let's see what else we have. Ooh, this is a really great comment, Krista, about us being made up of bacteria more than we are made up of cells. It's true that we are kind of a whole community of different kinds of cells, and some of those cells belong to different bacteria. It's kind of difficult for us to draw the line between, which bacteria are just kind of things that live in us and help us, and at what point do they become part of us. It's a really interesting idea. I like that. So what we're going to do is we're going to take a look at DNA on a much kind of further back scale, and we'll talk a little bit more about it once we get there. We're not going to be able to see this little ladder shape, that helix, that shape that we were looking at earlier, but we will be able to see kind of some clearish clumps when we do this experiment together. So what I've asked you guys to do is to grab some different kinds of plant samples that are easy to squish up. One kind of plant that is often used for an experiment like this is a strawberry. And there's actually a couple reasons for that. One of those reasons is because strawberries are really soft and easy to squish. So I'll start my experiment. The first one I'll do is on a strawberry. So I've got my strawberry, and now I need to make a solution. I need to make a liquid that will help me break down the cells of the strawberry to let the DNA out. Remember that picture we looked at earlier where all of the DNA was tangled up inside of that nucleus? We want to break it out so that we can see it. So what I have here is about a cup of water. You don't need all that much, or you can use more, but I'll show you kind of the ratio or the amount of other stuff we're putting in it. So I've got about a cup of water, and I've got a little bit of dish detergent. This is for washing dishes or washing other stuff. I'm just going to add a little bit here, kind of a big squirt like that. You don't need a whole lot. You're going to stir it up just a little bit. You don't have to stir a lot. We don't want to have tons of bubbles. This is a great point from Max here. If you're not doing the experiment along with us or you don't have all the supplies, that's totally okay. You can always take a look at how we do it and try it later. So I put in some of this dish liquid, and now I'm going to add a little bit of salt. This is just regular salt, and this isn't super exact, but maybe half a tablespoon or so. So I've added it in there, you can see it, and I'm going to stir it up a little bit. So this solution is actually what's going to help us break down the cell walls and free that DNA to come floating out, right? The dish soap helps kind of break up what we call the lipid layer, kind of the outside of the cell. So that kind of helps break it open. The salt is also something that kind of helps us rough up the cells and get them open. And our water is the solution, of course, that our DNA will be floating in. I have one more ingredient I'm going to get really fast because it's still in my freezer. I want to have some isopropyl alcohol or rubbing alcohol. I'm going to be using 91%, but you can also use a smaller kind of concentration. You can use a 70%. I'll be interested to see if there's any differences in the yield, the amount of DNA that we get out. But for this one, I'm going to be using 90, and I want to make sure that it's really cold. I'm going to be right back and grab my isopropyl alcohol. This has been in the freezer overnight. It doesn't have to be there for that long, but we want it to be there for about an hour so that it is nice and cold. Now, isopropyl alcohol, especially something like 91%, like this, doesn't freeze like water does. So this is still a nice liquid here, but it's really, really cold, frosty liquid. So I've got my solution, right? That's going to help me break down the cell walls. I've got my plant sample, right? What I'm going to do is I've already cut up a little piece of strawberry, and I put it in a plastic bag just to kind of keep the mess down, right? Now we said that we need to kind of mush up our plant sample. I'm going to be mushing up this strawberry, and to help me mush it up a little bit, I'm going to be putting in some of my solution. So just a little bit here. And this is not a super exact measurement, but I'm just going to give it a little pour there. We don't need a whole lot, especially because the cup I have is fairly small. So I've got about that much. I'm going to zip up my bag, and hopefully it will not leak. I'm going to try to get as much air out of my bag as possible. There we go. So I've got my solution here, and I have got my plant bit, and I'm going to start mushing up my strawberry. So I am mushing it up into teeny tiny pieces because I want to help kind of break down some of these cells, and I want to, while I'm doing that, I'm swishing it around in my solution, and I want to mush it till there are no more lumps if possible. So we want to get it super fine. I'll let you guys get a chance to do some mushing. Oh, this is a great question from Christina. I have not added alcohol yet. That is a great question. All I have added is my solution of water, dish liquid, and soap. So that's all I've got right now. And this is a good question from Christy about how much alcohol do you need? It depends. What you want to do is have maybe as much alcohol as half of this amount of liquid, right? When I pour this out, if it comes to about here in my cup, then I want to make sure to have about this much of the rubbing alcohol. And I'll show you what that looks like when I pour it. We have someone who commented, strawberries are delicious. They are so yummy. This one, of course, is very salty and soapy, so it might not be super good, but I actually can still kind of smell it. It smells nice. So I have been mushing my strawberry up really well here. And I'm going to switch camera views right now so that you can see my little beaker a little bit closer. So I'm going to go ahead and switch my camera here. We have a question about, can we mash the strawberry before we add it to the baggie? You sure can. That's totally fine. You just want to make sure that at some point you have your kind of solution, your dish soap and water and salt, and your mushed up fruit or plant sample all in the same place and getting mushed up together. So I've kind of got my sample here, and I've got a little cup, and I've got something else. I have a strainer, just a little mesh strainer like this. You can also use cloth if you don't have a strainer. We just want to make sure that we don't have too many chunks of strawberry or too many chunks of whatever plant we are using. So I'm going to kind of line this up here. And now I'm going to open just the corner of my bag, just a little bit, and I'm going to all right, sorry about that. My speaker likes to turn off sometimes. Thank you guys for bearing with our technical difficulties. This is the second lab we have ever done online, so we're still figuring it out. We really appreciate you guys being patient with us. So I have opened the corner of my baggie, and right now I'm just going to pour in my strawberries and solution and try not to get it everywhere. All right. So I have got some of my mixture here, and I'm just going to stir it up so I get as much liquid as possible in my little cup. You can use, I have a stir stick, you can use a spoon. I'm also going to use my hands. I can do that. This is okay to touch, right? We know that it's only made out of dish liquid and soap and water, so we know it's okay to touch. So I'm going to go ahead and put my strainer aside. I've got a little kind of bowl here of water to rinse it, but you don't need one of those unless you're going to do multiple experiments like I am. So I've got right now some liquid in here, and it looks kind of like strawberry lemonade, but if strawberry lemonade were made with like salt and dish soap and strawberry, right? Not very delicious, but it looks nice. It's nice and pink. And right now, all of the DNA is floating around in this solution, but it's not all attached, right? It's just these teeny, teeny, tiny pieces that are floating around. What we're going to do is we're going to make the DNA kind of chain together, because it's really good at that. It's what we call a polymer. It likes to hang on to itself. We're going to be adding our isopropyl alcohol now. Here's a big, big close-up of my alcohol bottle. And when you add it in, you want to make sure that you're not disturbing your liquid very much, right? I want to make sure that I'm pouring it in really carefully to the side of it. And again, we said I have about this much liquid. I'm going to pour in maybe about this amount of my rubbing alcohol. So what I'm going to do is I'm going to pour it in, and then I'll have you guys watch what's happening. Let's see if it'll focus here. So sometimes this is a little bit hard to see from the top, but do you guys see some of these little bubbles forming? So some of these little bubbles are formed around strands of DNA. So what I'm going to do is I'm going to show you kind of the side view. See how we've got kind of a separation going? Let's see if I can zoom it back a little bit. Here we go. See how I've got kind of a separation going between the top and the bottom? All that pink stuff is at the bottom, and the isopropyl alcohol and the DNA have floated to the top. Now, going to put my camera back down. Watch this. I'm going to take out some of this DNA. I'm going to use, what did I tell you guys to use? A fork. I'm going to take my fork and I'm going to take out some of this stuff. Whoa, do you guys see that? Do you guys see all that weird? It almost looks like boogers. We have all of this kind of stretchy, stringy stuff. This is the DNA of the strawberry. Now, strawberries are really, really good to use because they have a lot of DNA. They're what we call octoploid. That means they have 8 copies of their DNA. It's sort of like having 8 recipe books instead of just the 2 that we humans have. So they have a lot of DNA, and remember, all of the strands of DNA are microscopic, so this represents like millions and millions and millions of strands of DNA all clumped together so that we can see them. If any of you guys did the experiment along with me, can you let me know if you got any similar results? Were you able to get any DNA out? I've even got still a little bit there floating around. Sometimes you get a different amount of DNA. Ayla mentioned that it looks kind of gross. You're right. It does look kind of gross. But this was all inside of the strawberry, right? It was just locked up inside of its cells. Ooh, maybe we have some people who mentioned that they did get a good yield. And Max is, oh, repeating, that's perfect. The word octoploid, octo like an octopus. It has 8 copies of its chromosomes. So I've got a few more things with me here. And in the time I have left, let's see, maybe we have time for 1 or 2 more experiments for us to do. I've got some to choose from, guys. I've got, let's see, I've got some onions we could crush up. Those are really fragrant. I've got some kiwis, and my vote is for kiwis because they are what we call, well, they have 6 copies of their DNA. They don't have as many as strawberries, but we might get some good DNA from this one. I've also got some little bits of tomato. I've also got some banana. This one's good and squishy. And I've got some butternut squash here as well. I just picked some squishy stuff. What do you guys think? Can you vote really quickly in the comments? Say if you want to do onion or kiwi or tomato or banana. We have a vote for kiwi. Perfect. Thank you. Hexaploids. 6 copies. Ooh, we've got a different vote. So 2 for kiwis, 2 for banana, 3 for kiwis, tomato, banana again. So we're kind of between, let's say between banana and kiwi, because those are the easiest to squish, and we only have a few more minutes left. So I got 2 more votes for kiwi. So let's give that one a try. And remember, these are pretty commonly found plants or fruits, vegetables, stuff like that you guys may have in your home, or possibly you might have a little extra next time you go to the grocery store. So you can always do this experiment on your own. What I'm going to do now with my kiwis is I'm going to crush them up a little bit. And I can use my hands. I already kind of cut them into little pieces. But I'm also going to use something that I have here. This is called a billet. And this is not really for cooking, but it can help me for cooking in this case. So I'm squishing up my kiwis pretty good. I haven't put in my liquid yet, but I'll go ahead and do that. I just wanted to make sure I could squish them a little bit first. All right, so these are looking pretty good and squished. I'm going to add a little bit of my liquid here. Here's about as much as I have added. I'm going to close it up and squish it up. Can you guys tell me in the chat if you have done this experiment with me? Did you use a certain plant? Did you use a certain plant or fruit or vegetable or anything like that? While I'm squishing up this kiwi a little bit more. You can see all the little seeds in there. Oh, this is really cool. So someone, Christina did the strawberry and also did some lettuce. That's really interesting. So the strawberry worked really well. The lettuce, she says, didn't work as well, right? So the lettuce is a little bit harder to kind of squish up into pieces is one of the things, right? If we wanted to, take some scissors or a knife and squish it up teeny, teeny, tiny, we might get a little bit more. I'll give you guys one more hint at the end that might help when you do this experiment on your own if you want to. But first, I'm going to we're going to see how much we get from the kiwi. So I have pretty good crushed up kiwi. I'm going to get my strainer again. I rinsed it a little bit. Might still have a tiny bit of strawberry on it. Oops. All right. I'm going to pour my kiwi in. Try not to get it everywhere, but no promises. There we go. That looks good. So I've got my kiwi here. I'll mush it down a little bit with a fork, get as much of that kiwi liquid as I can. There we go. And now I've got this beautiful green solution here. This is my kiwi and salt water and dish soap lemonade here. So I'm going to go ahead and add my cold isopropyl now. Ooh, that's a really good question. I'm going to go ahead and pour this in, and I'll answer that question. We can watch and see if this one works the same as the strawberry did. So somebody had a great question about strawberries are octoploids. They have 8 copies of their DNA. Kiwis are hexaploids. They have 6. Humans usually have 2 copies. So we are diploid. And a lot of different plants are also diploid, but many of them are also polyploid, meaning they have more than 2 copies. So I'm actually seeing a little bit of that DNA there. I don't think it's as much as the strawberry. I'm going to see if I can get some out. Oh, there's a little bit. It's kind of hard to see here. There's a little bit on my fork. I'm going to see if I can show you guys the side view here. Let's see. Can we see that? It's a little bit murky. I can see some of those bubbles on the top, and actually if I gave it a little bit more time, it might help it to kind of congeal, clump up a little bit more. But I can see that I've got a few little pieces here. Ooh, there we go. There's one. A little piece there. See that kind of stretchy part? That's the DNA. Now there's not as much as there was with the strawberry, right? And part of that may be that I didn't mush it up as much. Part of it may be that the kiwi just has fewer copies of DNA. There's a lot of different factors, right? And we could test a lot of these different things, but this is unfortunately all of the time we have for today's lab. Really quick before the end, I'm going to switch my video back to me. Ooh, this is a really good question about copies of DNA that dogs and guinea pigs have. That's a really good question. That's one that I would have to look up. I just want to give you guys one more hint, kind of a my life hack here for getting more DNA out of your samples. I've got a little bit of pineapple juice here, and pineapple juice has some enzymes in it, so it's really good at breaking stuff down. If we use a tiny little bit of pineapple juice in our sample, like maybe in our kiwi, it'll help break open those cells a little bit more. Now, will we get a little bit of pineapple DNA in our sample? Maybe a teeny tiny bit, but this is pineapple juice, so there's not a whole lot of that pulp in there. I've poured a teeny tiny bit into my beaker here. Do a little bit more just before we go here. And actually, I think that may have helped a little bit. I'm gonna see if you guys can see this. I can kind of see it from the top. There's these teeny tiny strands that are on my fork now. You see those? Those are some of the DNA, and pineapple enzymes help me get those out. So that's something for you guys to try if you would like to. Unfortunately, that is all the time we have for today. Thank you guys so much for joining us. You had such amazing ideas and questions, and this was really fun to have you guys here. I know that I'm going to be interested in doing some of these other experiments at home. I hope you guys try it as well. I hope that you guys can join us next week, next Wednesday, when we are going to be making a kind of bioplastic from milk. That's going to be a really fun one. It's one of my favorites. So I hope I will see you guys all there.